Hello, I am Mason Cipollini, joined by Colin Brown and Leif. This is Lab 6 for OPTI 471B.
To understand this experiment, we first need intuition for what we are measuring, coma and astigmatism, which are off-axis aberrations. Coma can be understood as a pupil-dependent magnification. Rays from the same field point but different pupil locations form images at different heights, producing a skewed comet-like blur. As shown here, the image height depends on pupil coordinate and scales with pupil position, field height, and field angle. An example is shown below. To analyze coma, we use a transverse ray intercept plot, which shows where rays from different pupil points land in the image plane. Points A, B, C, and D represent different locations across the pupil, and P is the paraxial image point, which serves as our reference. First, consider rays in the tangential plane, which contains both the optical axis and the field point. Rays from pupil points A and B map to positions that are shifted upward relative to P, producing the elongated structure associated with coma. Now consider rays in the sagittal plane, which is perpendicular to the tangential plane. Rays from pupil points C and D map to a more compact region, forming the base of the spot. Together, these rays create the asymmetric distribution characteristic of coma. Is quantified using transverse ray aberration, measured from the paraxial point P. Here we see that the tangential coma is larger than the sagittal coma, with Seidel theory predicting a ratio of about 3 to 1. Astigmatism is another off-axis aberration, but unlike coma, it arises from differences in power between the tangential and sagittal planes. In other words, the lens focuses rays differently depending on the plane they lie in. For an off-axis field point, rays in these two planes do not come to focus at the same distance along the optical axis. To visualize this, consider an off-axis point that is vertically displaced from the optical axis. At a given point on the lens, the surface has different local radii of curvature in the tangential and sagittal directions, labeled here as RT and RS. Because RT and RS differ across the lens surface, rays in the tangential and sagittal planes experience different focusing strengths. This leads to distinct image formations depending on the orientation of the ray bundle. In this sense, astigmatism can be understood as a difference in effective power associated with different parts of the pupil, arising from the different curvatures in the tangential and sagittal directions. As a result, the system forms two separate foci, a tangential focus and a sagittal focus. Each corresponds to a line image formed by rays in that plane. The key result is that the image distances are different. This separation along the optical axis is the defining feature of astigmatism.
For the procedures, first align the laser to transmit through near and far pinhole. Place a surface into the beam path and mark the location of the beam for further alignments. Then, as in previous labs, position and translationally adjust the microscope objective until the output is focused on that marked point. Add the spatial filter and adjust both it and the objective until all fringes are visibly gone. Next, swap the spatial filter stand for a collimating lens and adjust until the retroreflections line up with the laser cavity. Once both these components are individually aligned, the beam expander can be assembled, where its collimation is verified with parallel fringes on a shear plate, as shown in Lab 4. Take the components off of rail 1 for now and add a mirror set upon a translation and rotation stage. Align the mirror with near and far pinholes, much like the laser. Keep a pinhole at the near location and place the focusing lens beyond it. Use that pinhole, like the laser cavity, to align the retroreflections of the lens. Finally, place the microscope tube alone on the rail and adjust until the crosshair and beam are aligned on the aforementioned marked point. With everything aligned, the full system is assembled with the Hartmann mask placed before the second lens. This component will be key to quantifying the aberrations. To do this, shine the reflected collimated beam onto the mask with no holes obscured. Move the lens along the rail until the resulting array of beams converge to a point at the entrance of the microscope tube. Using the micrometers, adjust Y and Z translations until all of the beams converge and are aligned with the crosshair on the marked point. This will provide the marginal focus for the lens. For each field, the following measurements were taken to obtain values for sagittal and tangential coma and astigmatism. First, all holes except the central is blocked, the microscope is adjusted along Y translation to the correct field position, and the steering mirror is adjusted such that the beam is recentered on the crosshair. Then, points A through D are selectively uncovered, and the translation on the Y micrometer required to recenter the beam is recorded. Then, the A, B, and C, D pairs are chosen, and both Y and Z translation is used to best overlap the spots.
For the ZMAX setup, an entrance pupil diameter of 1 inch was chosen based on our Hartmann mask. Unfortunately, we did not measure its size in the lab, and this is our best guess. It is a potential source of error. Then the lens data was entered, optimizing for spherical aberration, and the fields of -5, 0, and 5 mm image height were also entered. These are the values we took our data for. However, after finishing the lab, we learned it would have been better to choose an intermediate value, such as 3 mm instead of the symmetrical -5. A marginal ray height solve was used to find marginal focus. From our experimental data, the equations on the left were used to calculate the values for tangential and sagittal coma, with epsilon y values coming from the Y micrometers adjustments for points A through D. On the right are the Rayfan plots produced from Zmax for the various fields, where we use the same equations, with equivalent points A through D coming from the extremes of the XP and YP plots. For both fields 5 and minus 5, our experimental values are much larger than the theoretical ones produced from Zmax. For the minus 5 field, we found the expected ratio of 3 for tangential to sagittal coma, though the 5 mm field did not show the theoretical ratio. For sources of error, the spacing of the holes on the Hartmann mask was not known, which is a large source of potential error. Additionally, there were some slight misalignments in the optical system. While we did achieve the expected grid pattern while finding marginal focus, during later measurement steps, some of the Hartmann hole images were quite distorted and hard to determine their exact position. From our experimental data, astigmatism was calculated simply as the difference between the sagittal and tangential focus. For the Zmax data, we plotted the field curvature, which gave us curves for the sagittal and tangential focus, and the values at the extreme field, 5 mm, were used in the difference. Here, we find much better agreement with the experimental data for both fields.